The area of a colony is equal to pi r squared. Here's a Petri dish. Each of these orange blobs indicate a bacterial colony. You're expected to use this formula for the area of a circle to calculate cross-sectional areas of colonies. The area can be given in millimetres squared or in centimetres squared. Pi is equal to 3.14 and r equals radius. The radius could be given in millimetres or centimetres. So how do we calculate the area of a colony in practice? Find the area of bacterial colony A. So the first thing that we need to do is to measure the diameter of the bacterial colony A using a ruler. By doing this, we can see that A has a diameter of 10 millimetres. It's easier to measure the diameter first rather than the radius because the colonies can be really small. Step 2, halve the diameter to find the radius of the colony. So in this case, the radius is equal to 10 divided by 2, which equals 5 millimetres. So we have the radius. What do we do next? Step 3 is to insert the radius into the formula to calculate the area. So as a reminder, the area of a colony equals pi r squared. So this will be pi times 5 millimetres squared. The units for the area will be millimetres squared because we measured the radius in millimetres. In another example, this could be in centimetres squared. So let's move this calculation on. This is equal to 3.14 times 25. You can use a calculator to work this out. This gives us a value of 78.5 millimetres squared. So the area of colony A is 78.5 millimetres squared. You are expected to know how to carry out this calculation, and you could be asked to do this in the exam. So you need to memorise this formula and know how to apply it. The zone of inhibition is the area of clear space around an antibacterial substance in which bacteria are unable to grow. We can test different antibiotics or antiseptics and compare them to each other, or we could test different concentrations of the same to find the safest and most effective concentration to inhibit the growth of a particular bacteria. In our Petri dish, you can see these 3 blue dots. These are filter paper discs, and some of them are soaked in an antibiotic. Pure bacteria is spread onto the agar plate and the discs are added on top. When we get bacterial growth, we can see the zone of inhibition, that is, this clear space around the disc. The yellow colour indicates the bacterial growth, and this clear ring around the disc is the zone of inhibition. Investigating the effect of antiseptics or antibiotics on bacterial growth using agar plates like this is a required practical for your GCSE biology. So how can we measure how well an antibiotic works using the zone of inhibition? The area of the zone of inhibition is equal to pi r squared. This is the formula for finding out the area of a circle, and we can use this to calculate the area of the zone of inhibition as a measure of efficacy. The efficacy of the antibiotic basically means how well it works in preventing bacterial growth. The area could be calculated in millimetres squared or centimetres squared, depending on the measurement that you take. Pi equals 3.14 to 2 decimal places, and r is equal to the radius. This will be measured in millimetres or in centimetres. This disc right here is the most effective antibiotic. You can see this because it has the largest zone of inhibition surrounding it. This disc at the top of the dish had no effect. It has no zone of inhibition surrounding it. Remember that the larger the area of the zone of inhibition, the more effective the antimicrobial is at inhibiting the growth of bacteria. Let's work through an example of measuring the zone of inhibition. Measure the zone of inhibition of antibiotic A. So step 1. Measure the diameter of the clear space using a ruler. Using the ruler, we can see that the diameter of A is 12 millimetres. It's easier to measure the diameter because the zone of inhibition can be really small and it's harder to measure to the centre when you're using centimetres, so millimetres are much easier. Step 2. Halve the diameter to find the radius of the clear space. So in this case, it's 12 divided by 2, which equals 6 millimetres. That's the radius of the zone of inhibition for antibiotic A. Now we've got the radius, what's the next step? Step 3. Insert the radius into the formula to calculate the area. Remember that the area of the zone of inhibition is equal to pi r squared. So for this calculation, the area equals pi times 6 squared. This equals 3.14 times 36, which is 113 millimetres squared. Remember that these units can be centimetres squared or millimetres squared depending on the units that you use for the radius measurement. In the exam, you could be asked to calculate the zone of inhibition using pi r squared, so make sure that you know this formula.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy to follow videos and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE biology course. See you there!